Let's now move to the chi-square test, which is quite a sort of common test. So I know that you have in other parts of the specification, right, like other tests like the t-square or the Pearson or Spearman correlation. Sometimes students, need a little bit of guidance, like how do I know which test to use depending on the exam question? Because the exam question will not always tell you, and in fact, usually will not tell you which test to use because they want to see that you can understand your statistics well enough to be able to tell them yourself what you'll use when. However, the chi-square is the easiest one because somewhere, somehow in your question, you are going to have observed versus expected results. And so this is the formula, which is given to you. You don't have to memorize it. So it tests whether the differences between the observed and expected results are due to chance, or if there is what we call a significant difference, meaning what? There's an underlying biological reason why we observe a difference, and we call that a significant difference. Let's look at what the null hypothesis is and how we test it. So the null hypothesis states that there is no significant difference between observed and expected. So whatever difference we see is due to chance, and it will usually be a slight, a subtle difference, right? Chances are if you have a really big difference, it's most likely not due to chance. There's an underlying biological reason. However, you will have to prove that using the chi-squared. So if the null hypothesis is rejected, if the null hypothesis is accepted, then yes, we didn't have a significant difference between observed and expected. If it's rejected, however, and we're going to see exactly how that works, then the results are significant, or they have a significant difference between observed and expected due to an underlying biological reason. So we have two columns here. One column is the degrees of freedom. And how do we find the degrees of freedom? It is going to be the number of categories minus 1. What are the number of categories? Basically, they're going to be the 4 different, or the 3 different, depending on the precise cross, the number of different phenotypes that I'm expecting. I'm just going to write it here. Number of different phenotypes. In the case of genetics, right? Okay, so I will have my number of phenotypes, and that's how I'm going to know which row, to look at. How will I know which column to look at? We usually use the 0.05 significance level, which means that what? If there is a 95% chance that the null hypothesis can be rejected. So if the calculated chi-squared value is greater than the critical value at 0.0, what's the critical value? So for example, let's say that I've done my chi-squared test and I came up with a chi-squared value, for example, of, 10. And let's say that I've got 4 degrees of freedom. So I look up this value here, 9.49. If my calculated chi-squared value is larger than this value, then I reject the null hypothesis. If my chi-squared value is smaller than that, then I accept the null hypothesis, meaning that there's no significant difference. And we're going to see this as an example. Don't worry. I'm going to practice it. So a student conducted genetic crosses with sweet pea plants. He tested his results for statistical significance using the chi-square test, obtaining a result of 7.61. This experiment had 3 degrees of freedom. Using table 1, state whether or not the student is able to reject the null hypothesis and explain why. So pretty simple. So my context here is the chi-square test, and my command is to state and explain. My direction is that whether he accepts or rejects the null hypothesis. So he cannot reject the null hypothesis since the value that he calculated is not greater than the critical value of 7.28. If I look at my table, 7.28. Sorry, where is it? A2. The difference is not significant as there is more than a 5% probability that the effect is due to chance. There's 95% probability that the effect is due to chance. Let's go through like a walkthrough of exactly how we apply the formula of the chi-square test. So a student performs crossbreeding experiments with fruit flies, and she expects a phenotypic ratio of 3:1. She can use the chi-square test to determine the actual results and whether they fit the predicted ratio. So here are the observed frequencies or numbers of the phenotypes. How am I going to work out the expected? So I know that the expected ratio is 3:1, but how will I work out the actual numbers of individuals of the expected ratios? Well, I'm going to add these two up, which add up to 100. And those 100 total individuals should be distributed in a 3:1 ratio, right? So basically 75 of them should be this phenotype and 25 should be the other phenotype, which adds up to 100. This is what you're always going to do whenever you're given a question like that. If they give you the observed phenotypes, you're going to add them all up and get the total. And then how do I calculate this 75? So basically 100 times, divided by 3 is 25. So 3/4 basically of 100, which is 75, is going to be this phenotype, and 1/4 is going to be the other phenotype. So then I now go ahead and I calculate my chi-squared step by step, right? So this is, of course, observed. This is expected. This S stands for sum of. So the best way to do it is to really create these columns and to be able to calculate each term separately. So first I calculate my observed minus expected. So observed minus expected here is 7. Observed minus expected here is -7. It's okay to get a negative value because it's always observed minus expected, not the other way around. I now square these, so it's now 49 for both. And finally divide those with the expected values. So I'm going to get 0.5653, 1.960. And so then my chi-squared value comes out to be 2.613. Got the answers ready here, so I'm not actually using a calculator. Just going to give you a second to jot this down. What do I now do with this value though? Okay, I've got my chi-squared value. How will I determine whether I accept or reject the null hypothesis based on that? So I, of course, have to go and use my chi-squared table, which doesn't seem to be here. So how do I use my chi-squared table? I need to know the degrees of freedom, which is number of categories minus 1. What is the number of categories here? Well, it's my 2 different phenotypes, right? So degrees of freedom here is going to be 1. If the chi-squared value that I calculated, the 2.61, is smaller than the critical value, which if I read my chi-squared table, it's going to be 3.84 at the 0.05% significance level, then the null hypothesis cannot be rejected because this is smaller than 3.84. Sorry, what did I do? Okay. Therefore, the difference observed is due to chance and not due to an underlying biological reason. Let's have a look at this other example question. So a student performs a dihybrid cross with 2 heterozygous parents. The dominant allele for seed shape is round and the dominant allele for seed color is yellow. Number of offspring produced are shown in the table here. The student expects the usual phenotypic ratio seen in a dihybrid cross and assumes there is no autosomal linkage or sex linkage or codominance or epistasis. We're going to see about those later, but they're basically ways that one can deviate from the expected phenotypes. These are like the possible underlying biological reasons why I may reject the null hypothesis, right? Sex linkage, epistasis, codominance, we're going to see those. So let's look at our context and command word and so on. So what does the question want me to do? Perform a statistical analysis to determine if the observed results vary from the expected results. State an appropriate null hypothesis for 1 mark, determine the number of degrees of freedom, calculate the value of chi-squared, conclude as to whether the null hypothesis should be rejected. So let's break this down. What is my context? My context is basically the previous page, right? Previous page, as well as these numbers here. My command words are right here. They're fairly obvious. State, determine, calculate, conclude. Finally, my direction is that I'm going to perform a statistical analysis and I'm going to formulate a null hypothesis. I'm going to state the number of degrees of freedom, value of chi-squared, and whether the null hypothesis should be rejected. So let's take each one of those step by step. What is the null hypothesis? That there is no significant difference between the observed result and the expected results of phenotypes. How are we going to determine the number of degrees of freedom? Take a quick moment to consider that. What are the number of categories that I have? Here they are. 1, 2, 3, 4, the 4 different phenotypes that I'm expecting. Therefore, my degrees of freedom are 4 minus 1, which is 3. And finally, I have to actually calculate the value of chi-squared. It's quite a lot of work for only 3 marks, unfortunately. But overall, the entire question together is, of course, worth more. Again, I have the challenge of how will I figure out the expected ratios from the observed ratios. So I have to know, I am just told in this question that I have the that I am expecting a classic, I'm told. She expects the usual phenotypic ratios seen in a dihybrid cross, assuming no linkage or anything else. What are the expected phenotypic ratios seen in a dihybrid cross? Well, they're 9, 3, 3, 1, aren't they? In a 9, 3, 3, 1 ratio of expected phenotypes, what are the total phenotypes? If you add all of these up, they come up to 16, right? So the total number of individuals is 1,392, And out of those, they have to fall into that ratio of 9, 3, 3, 1. Meaning what? That I'm going to have the 9 bit of the ratio, 1, 3, 9, 2, times 9/16, right? 9 out of the 16 possible, out of the 16 total ratios. Then I'm going to have 9,392 times 3/16 twice because it's 3, 3. And finally, divided by 16. This is how I'm going to come up with my expected ratios. So the actual numbers that come out of that are, I'm going to write them in a different color so you can see them. So this comes out to 783. This is 261, 261, and 87. Then this is just like calculator, right? Then I come up with my observed minus expected, which is 21 minus 23 and minus 4. I square those. And divide them with the expected. These answers actually for the table, don't worry about jotting down all of those numbers. They will be sent to you. So my total then chi-squared value is 2.31. Okay. Now, how do I then decide whether I accept or reject the null hypothesis? Well, I know that I've got 3 degrees of freedom. So I'm looking at this row and I am looking at the 0.05 significance level. So my critical value of my chi-square test is 7.82. I then go ahead and compare it with the value that I calculated, which is 2.31, which is of course much lower than the critical value. Therefore, the null hypothesis cannot be rejected. So the difference, the deviation that we see between observed and expected is due to chance.